Human beings have always been dependent on animals such as cattle, birds, pigs, sheep, and bees for products like milk, eggs, wool, meat, and honey. With an increase in population worldwide, an increase in food production is also required. This can be achieved through animal husbandry and plant breeding. Animal husbandry refers to the practice of breeding and rearing livestock that are useful to human beings, and includes cattle or dairy farming, poultry farming, beekeeping, and fisheries. Plant breeding refers to the manipulation of plant species to create desired plant types that are better suited to cultivation, giving better yields, and that are disease resistant. Did you know that India and China together contribute more than 70% to the livestock population in the world? However, as the productivity is very low, their contribution to the world farm produce is a mere 25%. To increase the overall quality and productivity of animal produce, strategies and techniques to enhance food production need to be initiated, along with proper management of farms and farm animals. Let us look at the management practices followed in dairy and poultry farms, where traditional methods have been combined with innovative technologies to boost food production. Dairy farms rear animals for milk and milk products. The management methods used to increase production without compromising on the quality of milk is called dairy farm management. One of the key factors for increased milk production is breed selection. Good breeds have high yielding potential and high resistance to disease. Apart from the breed, proper sheltering, feeding, and cleaning of cattle are essential to obtain high yields of milk. Sheltering involves providing a clean and disease-free environment with sufficient ventilation and water for the animals. It is also very important to keep a check on the health of the cattle through regular visits of a veterinary doctor. Their feeding is carried out in a scientific way, involving the supply of fodder that is checked for specific quantity and quality. Cleaning the cattle and their shelters is also important to avoid any risk of germs and diseases. Apart from cleanliness, hygienic measures should be followed to keep the cattle and the handler clean. To avoid any contamination during milking, storing, and transporting milk from the farm. Nowadays, with new technologies such as milking machines that are used to harvest milk, most of these processes are mechanized, and the possibility of contamination is greatly reduced compared to the old method of milking by hand. In addition, the proper functioning of a dairy farm can be monitored by periodical inspection and record keeping. Just as dairy farms are used to rear milk-yielding animals, poultry farms are managed to raise domestic fowls or birds, such as chicken, duck, turkey, and geese. These birds are reared for their meat and eggs. In fact, Poultry refers to the meat of these birds and is sometimes used to denote the meat of all kinds of birds. Poultry farms also employ the management practices of dairy farming, such as proper breed selection, shelter, feed, and healthcare. Like milk, chicken and eggs are also consumed by human beings, which is how bird diseases spread to human beings as well. Bird flu or avian flu is a very common disease. If the chickens get infected by such diseases, they need to be properly culled and disposed of. However, ensuring proper hygienic shelter for the birds, nutritious feed, and regular health care, along with good quality breed selection, will result in an increase in chicken and egg production. In this manner, proper poultry farm and dairy farm management, along with strategies and techniques to ensure good yield and quality, will help increase the overall quality and productivity of animal produce. Animal breeding is the controlled propagation of animals to achieve desirable qualities of farm animals and increase the yield of these animals. The term breed refers to a group of animals within a species related by ancestors and possessing many similar characters such as appearance, behavior, and size. For example, Jersey and Leghorn are very popular breeds of cattle and chicken used for breeding. Animal breeding is divided into two main categories: inbreeding and outbreeding. Inbreeding is breeding between animals belonging to the same breed with a common ancestor, whereas outbreeding is breeding between animals belonging to different breeds, or between the same breed but with different ancestors, or between different species. Let us learn more about inbreeding. Animals selected for inbreeding are of superior quality and are closely related for 4 to 6 generations. A superior quality cow is one that produces more milk, and a superior bull is one that is a strong draft animal and gives rise to superior offspring. Subsequently, mating is carried out between offspring which have superior qualities. Thus, inbreeding increases homozygosity in animals. That is, it results in a pure line of animals with desired characters. This is similar to Mendel's experiment that yielded homozygous pure lines in pea plants. Inbreeding also helps in the expression of desirable characters and the elimination of undesirable characters in the offspring. However, inbreeding can increase the chance of expression of recessive genes, which are harmful. If inbreeding among closely related breeds is continued, it can result in inbreeding depression, which is reduced fertility and productivity. Now let's learn more about outbreeding. Outbreeding can be carried out in 3 ways: outcrossing, crossbreeding, and interspecific hybridization. Outcrossing is mating between animals of the same breed but with no common ancestors for up to 4 to 6 generations on either side. This type of breeding overcomes inbreeding depression. Crossbreeding is mating between animals of superior quality belonging to different breeds to produce hybrids possessing superior qualities of both their parents. For example, Jersey with a long lactation period and red Sindhi with high disease resistance are mated for a new breed with both the desired characters called Jersindh. The new breed obtained is then subject to inbreeding to increase the herd population. Another example of crossbreeding is the cross between Bikaneri ewes and Merino rams to form a new sheep breed, Hisar Dale. The third type of outbreeding is interspecific hybridization, where animals of two different species are mated. For example, mating between a male donkey and a female horse to form a new breed, a mule. Although most new breeds are infertile, they have high economic value. Both inbreeding and outbreeding can also be carried out using controlled breeding experiments. Artificial insemination is one such method. In artificial insemination, semen is first collected from the superior male. The semen is then injected into the reproductive tract or vagina of the female animal, or can be frozen for future use. Thus, artificial insemination is carried out to overcome problems in natural breeding and also to carry out mating between animals that are geographically separated. However, the success of animal breeding using artificial insemination is low, and so new techniques like multiple ovulation embryo transfer technology, or MOET, were developed. MOET involves the administration of gonadotrophic hormones like FSH and LH to female animals, which induce maturation of follicles resulting in superovulation. Superovulation is the production of 6 to 8 eggs instead of 1 egg. The superovulated female is mated with a superior male either through natural mating or artificial insemination. This results in fertilization of all the eggs. When the fertilized eggs reach the 8 to 32 cell stage, they are non-surgically removed and then implanted into different surrogate mothers for further development in a process called embryo transfer. Now, the female animal can be used for superovulation once again. In this manner, MOET helps increase the herd population containing desirable characters in a short span of time. MOET is widely carried out in animals such as cattle, buffaloes, and sheep. So different techniques such as artificial insemination and MOET, along with various inbreeding and outbreeding methods, are employed in animal breeding to produce offspring with superior qualities. Animal husbandry includes beekeeping and fisheries, in addition to cattle and poultry farming. Beekeeping, also known as apiculture, is the rearing of honeybees in artificial beehives called apiaries for the commercial production of honey. Honey is highly nutritious and an important constituent of several medicines. Also, the apiaries in which honeybees live and nurture their young ones are a source of beeswax. Beeswax is used in cosmetics and polish. Due to the increasing demand for honey, beekeeping is done both on a small and large scale. In small-scale beekeeping, 1 or 2 apiaries are kept in verandas or on rooftops, whereas in large-scale beekeeping, many apiaries are used. As the quality of the honey depends on the pasture available near the apiaries, beekeeping is usually practiced near fruit plantations and cultivated fields. Honeybees act as pollinators by helping crops like sunflower during flowering. In turn, they get nectar from the flowers. As a result, farmers get a high yield in terms of crops as well as honey. The most commonly reared honeybee species is Apis indica, also called the Indian bee. Although beekeeping is easy, it does require some knowledge and training that is provided by certain organizations. Some tips to be kept in mind are knowing the nature of the honeybees, selecting a desirable location for the beehives, controlling swarms, managing beehives during different climatic conditions, and collecting honey and beeswax. Following these tips will help carry out beekeeping successfully. Like beekeeping, fisheries are another part of animal husbandry. And relates to the rearing, catching and selling of aquatic animals such as fish and shellfish. As fish contain more proteins and less fat, they are preferred to other meats. Most commonly eaten freshwater fish include katla, rohu, and the common carp, while marine fish include hilsa, mackerel, and pomfret. Fisheries also provide products such as cod liver oil, which is a nutritional supplement, and pearl oysters that have commercial value. Consequently, it forms the livelihood of many people in coastal areas of India. As seafood like fish, prawn, crab and lobster are always in demand, several techniques like aquaculture and pisciculture are employed to increase their yield. Aquaculture is the cultivation of aquatic animals and plants for food, whereas pisciculture refers specifically to fish farming. This dramatic increase in seafood production was called the Blue Revolution, compared to the Green Revolution that brought about an increase in agricultural production. India is an agricultural country with 62% of people engaged in agriculture for their livelihood. With the passage of time and an increase in population, the demand for food has risen extensively. Earlier, this was tackled through traditional farming and better management practices. However, it resulted in a very small increase in yield. It was only during the 1960s that the Green Revolution brought about a drastic increase in the production of high-yielding, disease-resistant crops like wheat and rice by employing plant breeding techniques. Plant breeding involves crossing between plants with desired characters to produce offspring that possess the superior characters of both their parents. It dates back 9,000 to 11,000 years ago, when conventional plant breeding was practiced. Conventional or traditional breeding is crossing plants for desirable characters sexually through self or cross-pollination, or asexually through grafting. Most of the major crops such as wheat, rice, and maize are the result of conventional breeding. On the other hand, classical breeding uses techniques such as mutation or tissue culture, followed by crossbreeding of pure lines and artificial selection of plants. The desirable characters of plants generated by plant breeding include high yield with quality, tolerance to different types of environmental stress, resistance to microbes, and pests. Plant breeding is commonly carried out in government research institutes and some private organizations and involves several steps. The first step in plant breeding is the collection of germplasm of a particular crop that is intended for breeding. Germplasm is the collection of all the genes of different varieties of a particular crop in the form of seeds or plants. In this way, the naturally available gene pool can be utilized for breeding. The second step is the selection of superior parents. Here, two plants with the different desired characters are selected from the available germplasm. Then, they are made to self-cross to get a pure line of those varieties. The third step is cross-hybridization of the superior parents. This is done by collecting the pollen grains of one parent plant and placing them on the stigma of the other parent plant. However, the hybrids obtained may not contain all the desired characters present in the superior parents. In fact, 1 in 1,000 or 2,000 hybrids obtained may contain the desired characters of both superior parents. Hence, this step is considered cumbersome. The fourth step is the selection of superior hybrids. The desired hybrid is selected from the thousands of hybrids formed during crossbreeding that possess the characters of both its parents. Hence, it is considered superior than its parents. The hybrid is then allowed to self-pollinate till it forms a pure line of homozygous plants. The final step is the evaluation of the hybrid for the characters gained. This is done by first cultivating the hybrid in a research field under ideal conditions. Later, they are cultivated in farmers' fields at different locations for 3 growing seasons. This helps in comparing the performance of the hybrid against the best crop cultivar available in that area. In this way, plant breeding has increased the yield of many crops, including wheat, rice, sugarcane, and millet. The Green Revolution is a result of using various plant breeding techniques to boost agriculture. During this period, there was a dramatic increase in the production of wheat from 11 to 75 million tons and rice from 35 to 89.5 million tons. The increase in production was credited to the semi-dwarf varieties of wheat and rice that were developed. The semi-dwarf wheat variety was first developed at the International Center for Wheat and Maize Improvement in Mexico by Nobel laureate Norman E. Borlaug. Likewise, the semi-dwarf rice variety, IR8, was developed at the International Rice Research Institute, IRRI, in the Philippines. Later, better-yielding varieties of wheat hybrids such as Sonalika and Kalyan Sona and rice hybrids such as Jaya and Ratna were developed in India. In the case of sugarcane, Saccharum barberi and Saccharum officinarum were crossbred to get a hybrid. The hybrid obtained had both the characters of its parents, such as a very thick stem with higher sugar content, and the ability to grow in northern parts of India. Similarly, high-yielding hybrids of millet, such as jawar and bajra, which are resistant to water scarcity, were also developed in India. In this manner, through various plant breeding techniques, many high-yielding crops were developed that helped boost Indian agriculture. Plant breeding is the skillful use of different plant varieties to produce a desired plant type, thereby improving crop yield. But in most cases, crop production is affected by several crop diseases caused by pathogens such as fungi, bacteria, and viruses. Blast of rice, red rot of sugarcane, and late blight of potato are examples of fungal plant diseases, while black rot of crucifers is a bacterial disease, and tobacco mosaic virus and turnip mosaic virus are viral diseases. In such cases, plant breeding can be used to develop crops possessing resistance to a particular pathogen, provided the causative pathogen and its mode of transmission is known. Resistance can be defined as the crop's ability to prevent a pathogen from attacking it, and is determined by the genetic makeup of a particular crop. The genetic makeup of crops can be altered using plant breeding techniques like conventional breeding and mutation breeding. Conventional breeding involves hybridization and artificial selection. It involves several steps that include collection of germplasm, selection of disease-resistant parents from germplasm, hybridization of the disease-resistant parents, selection of disease-resistant hybrids, and evaluation of the selected hybrids. There are many disease-resistant hybrid varieties of different crops developed through conventional breeding. Let's take a look at some of them. Himgiri, a hybrid variety of wheat, is resistant to fungal diseases such as leaf rust, stripe rust, and hill bunt, while Pusa shubra, a hybrid variety of cauliflower, is resistant to fungal diseases such as black rot and curl. There is Pusa komal, a hybrid variety of cowpea that is resistant to blight disease caused by a bacterium, while Pusa sadabahar a hybrid variety of chili, is resistant to viral diseases such as tobacco mosaic virus and leaf curl. The only drawback of these disease-resistant wild relatives is that they are low-yielding. So, the disease-resistant genes from these wild relatives need to be transferred to high-yielding cultivars. As the disease-resistant genes are available in the same crop species, the transfer of disease resistance genes between the crops is done by sexual hybridization. For example, the yellow mosaic virus-resistant variety of bhindi, Parbhani Kranti, is grown by the transfer of disease-resistant genes from its wild relatives. Although many disease-resistant hybrid varieties are produced through conventional breeding, the process is limited by the number of disease-resistant genes available in different crop varieties and their wild relatives. As a result, another plant breeding technique called mutation breeding is widely employed. Mutation causes changes in the base sequence within genes, resulting in a new character not found in the parental type and results in genetic variations. Mutation breeding involves the use of chemicals or gamma radiations to induce alterations in the genetic makeup of crops. The second generation of mutation-induced crops possess new characters that are absent in their parents. These plants are screened for disease-resistant genes and then artificially picked for breeding. An example of mutant breeding is seen in a moong bean variety that is resistant to the yellow mosaic virus and powdery mildew. Apart from pathogens, pests also reduce crop production. As a result, there is a need to develop crops that are resistant to pests as well by using plant breeding methods. These involve the same steps that are involved in the development of disease-resistant plants. Germplasm of cultivars or wild relatives are used as a source of pest-resistant genes. Insect resistance in crop plants can be attributed to the morphological, biochemical or physiological features of a crop. For example, hairy leaves on cotton make it resistant to jacids, while hairy leaves on wheat make it resistant to cereal leaf beetle. Also, smooth leaves and the absence of nectar in some cotton varieties protect them from bollworms. Similarly, solid stems of wheat protect it from stem sawfly, whereas maize is protected from stem borers due to its high aspartate and low nitrogen and sugar content. There are quite a few insect-resistant hybrid varieties developed through plant breeding. Pusa Gaurav, a hybrid variety of rapeseed mustard, is resistant to aphids, while Pusa Sem 2 and Pusa Sem 3 are hybrid varieties of flat bean that are resistant to jacids, aphids, and fruit borers. And hybrid varieties of bhindi, Pusa Savani, and Pusa A4 are resistant to shoot and fruit borers. In this manner, plant breeding techniques help improve crop production, as well as prevent the use of bactericides and fungicides that can be harmful. They help develop crops that are resistant to pests and pathogens and can assist in boosting food production. Plant breeding plays a vital role in meeting the food demand of the increasing world population. While more than 840 million people around the world lack daily meals, it was reported that more than 3 billion people are fighting hidden hunger. Hidden hunger refers to a poor diet or malnutrition, that is, a deficiency of essential micronutrients, proteins and vitamins. As poor people cannot afford fruit and vegetables that are rich in nutrients, they survive on a diet containing staple food crops such as wheat, rice and maize. However, the level of vitamin A and micronutrients such as iron and zinc is very low in such crops, which makes them vulnerable to deficiency diseases that affect physical and mental health. To overcome this problem and promote public health, initiatives like biofortification were introduced. Biofortification is selective breeding of crops with higher nutritional value, such as proteins, vitamins, and minerals. For example, Atlas 66, a hybrid variety of wheat rich in protein, was bred with native wheat crops to fortify them. As a result, biofortification results in high-yielding crops fortified with specific nutrients. Another example of a biofortified staple crop is an iron-fortified rice variety that is 5 times richer in iron compared to its native variety. Similarly, maize hybrids containing double the quantity of amino acids compared to the other hybrids was developed. Did you know that the Indian Agricultural Research Institute, New Delhi, used biofortification to develop several fortified fruits and vegetables? Examples include vitamin A fortified carrot, spinach, and pumpkin, vitamin C fortified tomatoes, mustard, and bitter gourd, protein fortified beans and garden peas, and iron and calcium fortified spinach. Despite biofortification and several plant breeding techniques to boost food production, the demand for food was ever increasing. The shift to a meat diet further increased the demand for grains, as food grains are also used as animal feed. For example, a cow needs to be fed around 7 to 10 kilograms of corn to get around 1 kilogram of meat from it. For this reason, in addition to biofortified foods, scientists proposed the use of microbes as a food and feed supplement. These single-celled microbes are rich in proteins and are referred to as single-cell proteins or SCP. And so they are cultured to be used as a rich source of protein food. For example, spirulina, a blue-green algae, is cultured for its high protein and mineral content, and it is also a source of vitamins, carbohydrates, and fats. It is used as a food supplement for human beings and as a feed supplement for animals. Moreover, it helps reduce environmental pollution, as it can grow on wastewater from potato processing plants, straw, molasses, animal wastes, and sewage. Another example is Methylophilus methylotrophus, a bacterium that can produce 25 tons of proteins. Nowadays, fungi, such as mushroom, are accepted as edible. Scientists believe that soon, microbes too will be accepted as a food supplement. Tissue culture is a method used for plant propagation in addition to other traditional plant breeding techniques. It was during the 1950s that scientists found that any part of a plant grown in nutrient media under sterile conditions can give rise to a whole new plant. This property of plant cells to give rise to a whole new plant is called totipotency. Tissue culture uses this property of plant cells for asexual plant propagation in an aseptic environment. It requires a nutrient medium that contains sucrose, inorganic salts, vitamins, proteins, and growth regulators such as auxins and cytokinins. The tissue culture technique involves several steps. The first step is excising a small part of plant tissue like the root, shoot, or leaves of a plant. Then placing the cutout part or explant in a suitable nutrient medium, followed by allowing the explants to multiply and form a group of tissues called callus. The callus is then allowed to multiply to form many plantlets, which are then transferred to a different container for rapid multiplication. Finally, the developed plantlets are transplanted to the soil for normal growth. Therefore, tissue culture is performed using a small part of a plant to develop many plants, and so is also called micropropagation. As all these plants are developed from the same part of the parent plant, they are genetically identical and called somaclones. Micropropagation or tissue culture is employed in the large-scale production of tomato, banana, and apple. This technique can also be used to propagate high-yielding, disease-resistant varieties in a short span. Even if a plant is diseased, tissue culture can be used to get a healthy plant using the explant of the apical or axillary meristem. This is because the meristem is always free of disease and microbes. Culturing of meristems has been successfully carried out in plants such as banana, sugarcane, and potato. In addition to asexual propagation of plants, tissue culture is also used to mix two different species of plants that are otherwise sexually incompatible. Here, individual cells are isolated from each plant species and are placed in a culture medium. The cell wall gets digested in both cells, which aids fusion of the two protoplasts of different species, resulting in a hybrid protoplast. The hybrid protoplast is called a somatic hybrid, which gives rise to a whole new plant and may contain the desired characters of both species. This process of developing a hybrid plant using somatic cells is called somatic hybridization. It was employed while fusing the tomato and potato plants to obtain a hybrid plant. As a result, the hybrid plant resembled the tomato, but it didn't combine all the desired characters as expected. In this manner, Tissue culture, somatic hybridization, and other plant breeding techniques provide a huge potential for manipulation of plants to produce new varieties, boost